Thank you very much, and hope everyone can hear me. And we're going to dive into this, I guess, right away, and to cover some topics on CYP17 inhibition. These are the objectives that were circulated, no disclosures, and this is our outline. We'll discuss a little bit of the background to give us a bit of a framework going forward. We'll discuss some of the issues on androgen synthesis in prostate cancer, particularly CRPC. And then we'll discuss some of the current and new inhibitors which are in development listed there. Abiraterone we know about. Ortonel is in phase 3 trials. Glatterone and VT464 is the molecule which I was working on, and it is now in phase 2 clinical trials. So just to go way back, of course, androgen sensitivity in prostate cancer is credited to Charles Huggins and Hodges, and he won the Nobel Prize in 1966 for his work showing that prostate cancer is dependent on androgens and castration. Some of the work done here by Nick Brukovsky and others showed that DHT is actually the main active form of androgen in the prostate, and that there's an enzyme which converts it from testosterone. Discover the androgen receptor is another key background molecule, of course, in this pathway, and that was discovered in the 60s and 70s as well. So androgen synthesis in the body, of course, occurs in two places, in the testis and the adrenals. The Leydig cells stimulated by LH produces the vast majority, and then also in the zona reticularis of the adrenal gland. Now, I know that most of you know this off by heart and probably have it on your wall somewhere at home. And so we'll just go through this very quickly, but for those of you who just need a quick refresher, this is a steroid synthesis pathway. It's the same in the whole body, but the expression of the enzymes where it happens is not uniform. That's why it only happens in the testis and adrenals, because those are the places where the enzymes are synthesized for that. So it starts on the top left with cholesterol, and it goes through all these various intermediates, and it can go different directions depending on the expression of the enzymes in the tissue. It can go to androgens there, which is our interest here, and these are the different androgens. Of course, DHT is the most potent one in the prostate, but it can also be shunted through different pathways to estrogens, glucocorticoids, mineralocorticoids. Now, it's a pathway largely because there are several irreversible steps in the pathway, highlighted here, and these are the 5-alpha reductase, the P450 enzymes, including ours of interest here, CYP17. And now the CYP17 enzyme is unique, we'll discuss briefly, but it catalyzes two reactions, the 17-alpha hydroxylase and then the 1720 lyase reaction, and this occurs at different places here. There are also cofactors, particularly for the lyase reaction. There's something called the cytochrome B5, which is an important cofactor as well for that reaction. The other reactions here, which are reversible, exist in equilibria, are more variable in their expression, and that lends to different routes of going around things to get back to different substrates such as testosterone. So this is the enzyme. It's in the endoplasmic reticulum, part of the cell, and yes, it does result in congenital adrenal hyperplasia. It's one of many, I understand. And importantly, it blocks different pathways to DHT, both the classical pathway outlined here and also the backdoor pathway, and so it's a key enzyme in the biosynthesis pathway. As I mentioned, it's a dual enzyme with both hydroxylase and lyase function, and has cytochrome B5 as a cofactor, and also in humans it's been reported that it has other functions, including 16-alpha hydroxylate function, but that's not known to be clinically important at present, at least. Now, the very first CYP17 inhibitor is historical largely now, ketoconazole. It was originally it is an antifungal drug that was invented actually by Janssen in 1976, and they were testing it out at Stanford University. And these clinicians were very astute because they noticed in one patient that they had gynecomastia, and this actually tipped them off to ask why do they have gynecomastia. And so they thought about this, and then they had a second patient a year later, and then they rounded up some volunteers. Now, the volunteers they used was, in the spirit of medical tradition, according to them, was themselves and their postdoctoral fellow. And so they gave themselves ketoconazole and then measured their testosterone levels at various points and found out that it was decreasing after the dosage of ketoconazole. And they did some more in vitro experiments and and the rest is history, as they say. They presented their work at a forum in Chicago, and one of the fellows who was at this forum took note of this, went back to his institution at Johns Hopkins and talked to one of his urology fellows there and said, we have this drug that rapidly decreases testosterone. And so some of you probably know him, and together with William Pont, he published the first report, Dr. Trachtenberg, of use of ketoconazole in prostate cancer in 1983. Abiraterone was discovered in the 1990s at the Institute of Cancer Research, and as Mike German, they were looking essentially for a better molecule in the lab that had better pharmacokinetics and potency compared to ketoconazole. And so they did identify one, but there was some pushback a little bit because it was not called castrate-resistant prostate cancer at the time. It was called hormone refractory, and to have hormone therapy for hormone refractory prostate cancer didn't sound very attractive initially. But eventually it did go into clinical trials in 2003, and we know that it's now approved. So how does androgen synthesis in castrate-resistant prostate cancer occur in the body? This is a good review written recently, and you can see the different areas here. Particularly adrenal androgens are inhibited and intra-tumoral androgens. Not as much testicular androgens because they're usually castrated at this point. So again, here's the sources of androgens. In general, we also added here tumoral androgens, which can be intracrine or paracrine. Now, in CRPC, as I mentioned, we have no testicular androgens because they're on an LHRH agonist or they've been surgically castrated. And so the source of androgens is largely from the adrenals. They're high circulating levels. These can be converted into testosterone and DHT. And so this is not a new concept. Again, it actually goes back to Huggins' work in Chicago. Found that, or he suspected that adrenal androgens may contribute. And this was the long history of complete androgen blockade in the 90s of Fernand Labrie, and historically it was things like bilateral adrenalectomy and a glutathione, which also inhibits the steroid synthesis pathway, which all the residents will know exactly what step. And so these are the sources. So this is a review here showing your cortex of your adrenal, and you have tissue-specific expression of different enzymes. Here's cytochrome B5, which is your cofactor for CYP17 being expressed. This is a sulfosyltransferase enzyme, which gives you DHEAS, and but you don't actually have 3-beta HSD here, but the diffusion of molecules lets it happen here. And so again, here's the same thing, showing a tissue-specific expression of these enzymes in the zona reticularis gives you DHEA, DHEAS, and androstenedione. And so what is the relationship in prostate cancer? Well, we don't actually know if there's any clear relationship between serum and tumor androgens during ADT or even during CRPC. And it's not clear that the androgens in the blood actually increase during CRPC. There is some new data that's come out suggesting that your nadir serum testosterone on ADT may be predictive, but where that testosterone is coming from, whether the tumor or residual androgens, is not really understood. This is one study. This is a phase 2 study of abiraterone. They did subsequent analysis of 54 patients looking at what are the markers of response to abiraterone. And interestingly, they found that serum adrenal androgens predicted greater than 50% PSA decline. Now, I haven't seen this validated in the COU1 and 2 studies of abiraterone, but there is this study in the CO302 study. This is again pre-chemotherapy patients treated with abiraterone plus prednisone versus prednisone alone, and they looked at serum androgens and wanted to see again if this is predictive. And they found that if you had a good response to abiraterone and that your serum androgens dropped, that was associated with a response, PSA response, but it wasn't a complete correlation. So not all patients who had undetectable androgens had a PSA response, and vice versa. If you had a PSA response, didn't necessarily mean that your presumably adrenal androgens went to undetectable levels. So the heterogeneity is certainly a factor here. This is the data here showing that these are serum testosterone, serum androstenedione, and serum DHEAS. And when you give ABI plus prednisone, you get a huge drop in these levels. And actually you get a drop with prednisone as well alone. But this was heterogeneous, and it didn't clearly predict what happens. So there's definitely more going on. So adrenal androgens certainly play a role in CRPC, but it doesn't appear to be the whole story. And so we move on to intraprostatic or intra-tumoral androgen production. And this is one study from Seattle. They gave patients ADT, and then this is neoadjuvant, so they measured their prostatic DHT, and they found that it decreases as expected, but it decreases to a larger extent when you have benign disease. So if you have BPH, it really drops, and if you have Gleason 6, it drops by 85%, but if you have a higher grade cancer, it doesn't decrease as much, again suggesting that the tumor itself may be producing androgens. This is some of the work done by Jennifer Locke here and Martin Gleave several years ago, along with a group in Seattle, showing that there is intratumoral androgen synthesis. And they measured serum, this is tissue from metastatic bone deposits, and found that the testosterone level was actually quite high, and it was even higher than benign or localized prostate cancer in men who were uncastrated. And so the idea of androgen synthesis was clearly established in the lab. On the right is the LINCAP model, and these are the expression of the different enzymes, which we've all memorized by now. And they show that essentially when you get to CRPC, they increase over baseline and over their nadir, and this is the serum levels here. And so all the enzymes are going up, and the actual testosterone levels are going up in the tumors. It's a more recent study out of Seattle as well. And here they fed mice different diets which had more or less cholesterol. And essentially the more cholesterol they ate, the more they had in their serum. And if you look on the left here, on the bottom left, this is the different levels of serum cholesterol. And that, as that increased, your tumor testosterone increased, suggesting that they're synthesizing androgens not just from adrenal androgen precursors, but from cholesterol itself. This is from the same paper, again showing a correlation between cholesterol levels in the bottom and tumor weight, tumor testosterone, tumor DHT, and even expression of your CYP17 enzyme. And so that, again, suggests the importance of de novo synthesis. This is another slightly older, two different studies looking at gene expression. Here, these are untreated primary prostate cancer tissues in eugonadal men. And then these are metastatic bone deposits in this case, and not in this case. But here you see no difference between the levels of CYP17. There is more CYP11. And in this study, just highlighted over here, you do see an increase of CYP17. So, and these are essentially the same comparisons. The only difference is that these ones were laser microdissected, which means that you only had the tumor sample, where these ones took through the whole gross tumor from autopsy and homogenized it. And so there's a difference why perhaps other factors we'll get to in a few slides. But certainly it's a bit controversial whether they actually increase in humans or not. It's not clearly demonstrated. Another different study here looking at the same thing, gene expression of the different enzymes. Of course, we know the androgen receptor increases, highlighted by the red here. These ones all decrease, and these are involved with inactivating DHT. These one, the 5-alpha reductase, we do know that the benign 5A2 associated with benign tissue decreases, whereas the isoform 1 increases, and these are all in metastatic tissue. I didn't highlight it here, but CYP17 didn't show any clear changes in this study. And so these are some of the common gene changes. Essentially, you get more SRD5A1, as well as other enzymes such as this one, and these are involved with degradation of DHT. And there's also 5A3. So you thought you learned there's type 1 and type 2 for finasteride and dutasteride, but there's actually a third type which has been characterized as well. And this is fairly recent, and it appears to be at even higher levels than type 1 and type 2 of the 5-alpha reductase. It's not inhibited by dutasteride, and it essentially catalyzes the same reactions here. And so this may also be another enzyme which is important in CRPC, and could be an additional.
Can we ask a question?
Yeah, of course.
The responses to CYP17 inhibition are pretty significant in CRPC, and yet because of the availability of type 1 and type 2 compound reductase inhibitors, they've also been assessed in CRPC, and yet responses have been almost anecdotal. Can you explain why that would be? Or at least not explain, but speculate.
Yeah, you certainly do get a large increase of testosterone when you give 5-alpha reductase inhibitors. So you're not. It's in eukaryotic men, but it's also in. There's another study I didn't include in the presentation where they gave different combinations, dutasteride, bicalutamide, Zoladex in pre-prostatectomy men, and essentially it does increase your intratumoral testosterone levels. Not DHT. DHT drops very, very low, but you still have testosterone around, so that's one type of ligand, although not as potent. That could be one reason, certainly why, because you do have other types of ligand that's accumulating. That would be my first hypothesis. But there are different ways around the pathway as well. So if you inhibit one step, like HSB D6, I believe it is, also has a back conversion to DHT as well. And so there's always sort of another route which may be active when you inhibit one. And I think that's still an area of exploration.
We just completed a neoadjuvant study of 4 center, one in Toronto, Vancouver, also Boston and Seattle, where in one of the arms we've included both CYP17 inhibitor plus
Dutasteride. Yeah. Okay. Yeah, there was a paper, but I took it out of here for the sake of time. But this is just one slide to touch on the paracrine role of CYP17. And as I mentioned, that may be one explanation for the different levels. And this, again, some work done here showing that CYP17 expression. This is a picture of prostate cancer, and essentially it's highlighting the fact that the dark stain of brown here is CYP17. It's actually appears like it's secreted at least, and so Emma Gunn's lab at the Prostate Center is working on this a bit, looking at excretion of CYP17 in exosomes as a way of altering the tumor environment and perhaps influencing androgen synthesis in paracrine tissues in the stroma. And so that's another possible avenue of exploration, which is not very well understood and certainly not very well published on presently. So just to summarize this part, again, there's essentially 2 pathways of synthesis. You can convert to testosterone DHT from adrenal androgens, and then you can synthesize it de novo. And then there's autocrine and paracrine sources of androgen synthesis. And for both, I think the relative importance is fairly unclear, and it probably just reflects the heterogeneity of patients and tumors. There's many other questions which are unanswered. What is the role of AR mutations, some of which are sensitive to precursors in the pathway like progesterone? AR amplification, how does this work after enzalutamide? The role of progesterone, glucocorticoids in CRPC resistance, and really a big question, of course, is who's going to benefit and can you pick these patients out? And then, of course, when do you give it? Later in the disease or earlier? All very open-ended, unanswered questions, I think, presently. This is just one study to highlight briefly, showing that a particular mutation in one of the enzymes, 3-beta-HSD1, it's a bit busy, but if you have a mutation here, essentially the enzyme is not degraded at the same rate by the proteasome. And so you have more enzyme and more synthesis, and therefore more DHT and more AR activation. And they showed nicely in this paper that this is one mechanism that may contribute to CRPC. Overall, it was quite rare. It was less than 5%, but it does nicely show how different mutations may affect this pathway. As far as abiraterone following enzalutamide resistance, there's 2 small case series, one by a previous postdoc here and another by one of the medical oncology fellows here. And they both show essentially a fairly low response. We're talking 8% or 10% had a reasonable response to abiraterone post-enzalutamide. So this is fairly early, but it doesn't show great success thus far. And so there's a need to understand better and to pick our patients better. Again, just another slide highlighting a large article that was published in Cell. This is a commentary on it, showing that in enzalutamide resistance, the androgen receptor normally activates your androgen genes and pathway, but they suggest, and there's some issues with this paper, but some good data as well, that in enzalutamide resistance, perhaps the glucocorticoid receptor activate some of these same gene pathways. And so there's a bit of a switch from the androgen receptor mediating a lot of the signal to the glucocorticoid receptor mediating a lot of the same signal. But this is controversial, and this is a different paper looking at abiraterone, and then they added glucocorticoids. So these patients did not have prednisone. They were just given abiraterone, acetate alone, and they, of course, all had mineralocorticoid excess as a result. And so they then gave them dexamethasone. But in both time periods, they measured their steroids. And just summarize this very quickly, the results suggest that dexamethasone may actually suppress synthesis of steroids following abiraterone treatment. And this is just one graph from the study showing that when they gave, in red here, the glucocorticoid, in these patients, they had a rising PSA, they were progressing, they gave them dexamethasone, and they actually dropped their testosterone levels, they had a PSA response. And so certainly dexamethasone does do something beneficial, and this may be specific for abiraterone. I just highlight this pathway again to show that when you block both of these with abiraterone, you get an increase in progesterone, and this may stimulate mutations of the AR, and when you give glucocorticoids, you essentially suppress all the levels, and that may be one reason why. So switching gears here for the final portion here, targeting CYP17, just to highlight, there's a lot more interest in this, a number of articles published. Ketoconazole, abiraterone, we talked about, I'll say a few words, and then we'll go over some of the ones that are in development. So ortinel, as I mentioned in phase 3, there's a trial recently reported at GeoASCO, glatiraone VT464, I'll show you some of my data, and we won't talk about the last one for the sake of time. So ketoconazole has no survival benefit, just PSA response, has issues with drug interactions and so on. These are the...
How do you know it has no survival benefit?
Well, there has been, I think, one study that looked sort of retrospectively to see if there's, but there's no prospective studies.
It hasn't been tested in a 1,200 patient randomized study.
No, it hasn't. No, it hasn't. Yeah, just retrospective, but none of those suggest so far. So I think most of you are familiar with this. Abiraterone shows a survival benefit. This is post-chemotherapy. In the pre-chemotherapy, just highlight, this did not actually reach significance. They're pre-specified limits, but certainly the trend was for a greater survival benefit. And it's a prodrug metabolized abiraterone, and it's an irreversible inhibitor. It has other properties as well, which have been examined preclinically. It may inhibit the 3B2 enzyme. It may actually act as an AR antagonist as well. And then, of course, it works better when you have a lot of fat in your diet in terms of absorption. This is one of the newer drugs here. This is a non-steroidal selective and reversible CYP17 inhibitor. So it has more selectivity for the lyase function. And then we'll go over briefly the phase 3 study recently reported. And there's 2 other ongoing phase 3 studies. One in the pre-chemo setting with prednisone, and then one in the hormone-sensitive metastatic setting without prednisone. So the question you might ask is, if it's selective, why are you giving prednisone with it? Because if it's truly selective, then you won't have that accumulation and you won't have that feedback mineralocorticoid excess, which is why you give prednisone. And essentially they said from previous studies that prednisone has a benefit, so why should we hold this back to these patients? And so they gave it in the studies, but not in the hormone-sensitive one that's planned. This is the design of the trial, essentially randomizing patients to ortinel plus prednisone versus prednisone in a 2-to-1 fashion, and their primary endpoint was overall survival. They did not meet the primary endpoint, although one of the secondary endpoints did show significant improvement in radiographic progression-free survival. So moving along quickly, sub-analysis suggested that in the non-European population there was benefit, perhaps because they did not have abiraterone and enzalutamide therapies available, sort of muddying the waters, but won't get into that too much. Glatiraone is another new drug, and it's in phase 1-2 studies. Preclinical studies suggest that in addition to CYP17, it also is an AR antagonist, has AR degradation properties. This is a cartoon from their website. This is showing that it's selective. So ortinel this is the ratio, much more selective compared to abiraterone. Glatiraone is 3.2. This shows that in the lab here we could show AR degradation, the top line, with greater doses of the drug. And this is the design of the trial. So this is the drug I talked a bit more about I was working on. I don't have the structure because I'm apparently have to wait till it's published to show you, and but it's a non-steroidal small molecule inhibitor, and it has great lyase specificity. So in monkeys, if you give the drug, you don't get an accumulation of progesterone like you do here with abiraterone. And so it's very selective. The ratio is 9.2 compared to this study 0.17. And so again, just to highlight the fact that it only blocks this part of the pathway and not the hydroxylase function. Comparing it to the other drugs, the more you get to the left here, the more selective it is for the lyase, and the higher up you get, or pardon me, the higher up you get, the more selective it is, and the more potent it is to the left. And so this is sort of where it fits in the picture off the graph here in that regard. And so we assess this in preclinical models. These are C4-2 or androgen-insensitive cells, and we have enzalutamide-resistant cell lines, which we developed and treated with this drug. AR transactivation, for those of you not in the lab, essentially this is how it works normally. Your DHT binds to your nuclear DNA at the AR promoter and synthesizes AR protein. You can add in a plasmid which has the luciferase gene, which when you add a substrate gives you light, and so you can measure the activation of the AR promoter. And so doing that in these three cell lines, we found a decrease with abiraterone in C4-2, not so much in MR49C, and to some extent in this other enzalutamide-resistant cell line. But of course, good for this very small drug company of 12 people. It appears that it's slightly better, or significantly better in this case, with their drug. And so we looked at PSA. Again, here we actually saw an increase of PSA with abiraterone. This is after 6 days. No change in AR for all of them, but a decrease with the drug at this time point. Similar results with mRNA transcripts, showing a decrease with the drug here in the last 3 bars for AR-independent genes. Not so much here, but certainly some hint of a greater suppression there. Just going through this somewhat quickly, I apologize. Here we measured actual tumor steroid levels in the cells. Okay. So here we measured the amount of DHT and testosterone, and it's very low with the drug. But to our surprise, it actually increased, and this makes not a lot of rational sense that it increased with abiraterone. And so why that is, not sure. This is all the other metabolites that we looked at. Androstenedione is part of the backdoor pathway. We do see perhaps this is related to HSP3 suppression, that when you suppress that enzyme you get less progesterone and you get more pregnenolone with abiraterone at the higher doses. And this is actually probably artifact from high levels of abiraterone. But why there's activation of testosterone and DHT is a bit elusive. But it's probably related to activation of other alternate pathways, or perhaps that the resistant cells, because they're cultured for 6 days, do not take up abiraterone in the same way. But it's still somewhat surprising, certainly. This slide is busy, but it's to show you that these different enzymes in the pathway, when you give the either abiraterone or VT464, increase. And the increase is much greater with the VT464 on the right. And I think you can see that. And this is 3 bars or each cell line, but again, the trend is fairly straightforward, that you get a greater increase the higher dose of the drug, particularly with this drug. And so this is probably feedback, but also may represent a mechanism of resistance. Looking in vivo here, so we have tumors growing in mice that are enzalutamide resistant, and we treat them with the drug. So it's a very aggressive tumor, and so the differences are not humongous, but certainly because they die after a month of treatment, which is quite short in the lab for mice. But we did see a difference, both for abiraterone and for VT, really indistinguishable benefit. This is a waterfall plot. It shows a bit nicer that there does appear to be a trend for a better response with this drug compared to abiraterone. We found out after the study that mice and rodents probably metabolize the drug very quickly after 3 weeks, and so the benefit tapered off. And you can see this clearly in the PSA response here, in that it worked very well, but here at the once daily dosing of the drug, there was really no benefit by 4 weeks, and it's probably because they upregulate their enzymes and metabolize the drug, so they essentially have very little drug on board after 3 or 4 weeks. But again, you see a trend for better results with the drug. The dose of abiraterone we used as well, highlight, was very, very high, twice what's been published in the literature. We did find a marginally significant benefit for cancer-specific survival for the VT drug over abiraterone in vehicle. In this setting, again, they don't last too long, these mice. It's a very aggressive model. It's a pharmacodynamic study where we gave the drug for 3 to 10 days and then collected the mice and measured different parameters. In this small setting, we did show that there appeared to be better PSA decreases, and again, it's probably because it's earlier on and the upregulation of the enzyme in the mice was not an issue in this setting, but certainly a clear heterogeneity response, both in terms of PSA and CYP17 expression. The drug level appeared to be slightly higher with our drug compared to abiraterone, even at a very high dose. Looking at the steroid, and this is important here, these are castrate mice, so they don't have any testicular androgens, and mice don't tend to express CYP17 in their adrenals, and so they don't have really any adrenal androgens. So these are probably all related to circulating tumor androgens, and abiraterone, both of them decrease testosterone significantly in the tumor, but there's a trend to greater decrease with the drug. And the precursors, progesterone and pregnenolone, as expected, if you have a specific inhibitor of the lyase function, you won't get accumulation of this, and that was the case. And there was less with the drug in both of these factors. And DHEA as well was noted like that. This is gene expression. It didn't turn out too well, and I put it in PowerPoint here, but essentially the higher up you go, the lower amounts you have, and so with the VT464 drug, there's actually a greater expression of steroid enzymes. Again, probably feedback. You have lack of androgens, and so they're trying to upregulate the synthesis. And because we know that the steroid levels weren't high, it's actually not resistance, not a mechanism of resistance. This is some other interesting data that we generated. And here we gave a synthetic androgen to mimic DHT to the cells and then measured transactivation. And so, as expected, enzalutamide is an AR antagonist, and it's going to drop very significantly the transactivation. But to our surprise, VT464 did the same thing in these C42 cell lines, and abiraterone to a lesser extent. TAC700 had no drop. So this suggests that it's actually antagonizing the AR, which wasn't known, and we tested our enzalutamide-resistant cell lines as well. So here you expect that enzalutamide will not change your AR transactivation because they're resistant, and that was essentially the case. TAC700 here had the same thing. It has no AR antagonistic effects, but both abiraterone and VT again showed some AR antagonism, which, if you look at the gray bar here, appears to be greater with the VT drug. And so this is interesting. Certainly these cell lines have a mutation, the T77A, and the enzalutamide resistant ones have another mutation, which is related to enzalutamide resistance. And so in both these cases, there is AR antagonism with the drug, which is very important when you think clinically testing this in patients with enzalutamide resistance. So we also looked for AR degradation, found none. The doses we used, when we upped the dose to much higher, we did see some AR degradation, but again, it's a very, very high dose. Tested this out in a different model. This is wild type AR, so there's no mutation. And here again, TAC700 or ortonel has no AR antagonism, but both abiraterone and VT464 do. So to summarize, there is potent selective inhibition of CYP17 lyase. It does appear more effective than abiraterone in our models. Further targeting of the AR pathway does seem viable in our models of enzalutamide resistance. And I think our experiments show clearly that the mechanism of action is related to further suppression of the AR pathway through suppression of androgen synthesis, but also some exciting data showing that AR antagonism is also another mechanism of action, and it works against both wild type and mutant AR. So this work has augmented the efforts to get a phase 2 trial of this underway, which I understand started in February. Certainly more research is needed to select patients, and certainly there's some, I think, work going on looking at different mutations of the AR and whether this is useful, and also steroid levels are being measured in the trial to see if that can be a predictive biomarker. I think the effect of glucocorticoids, as we got into earlier briefly, is very controversial, and the use of this selective drug allows you to better evaluate that in preclinical models as well. So I think we're nearing the end here. Just to summarize again, progress I think has been made significantly in CRPC. We understand the biology better, and this has changed our treatment approaches, whether it's understanding that CRPC is still sensitive to androgen therapy, to exploring new treatments targeting other pathways. I think patient and tumor heterogeneity is a very key issue, which makes progress difficult, and predictive biomarkers will be helpful with that, and it's being integrated into most phase 3 clinical trials now, from what I understand. We again went over Ortonel, one of the new drugs, which unfortunately failed its first phase 3 trial, but has tumor on the way. Galaterone and VT464 are farther behind in the pathway, but certainly have a promise for the next generation of CYP17 inhibition. I would like to thank those who helped me in the lab. Both I work in Dr. Gleave's lab and in Amina Zubidi's lab, and I couldn't do this work without their help. And again, thank you to Martin and Amina for being my supervisors during this time. I'll take any questions if you have any.